Microbio lab.
In some I'm being sent to work.
I mean, riskier. If there is another 3 hours, then I'm leaving.
You say that every day.
And you're always someone the first one inside.
Wait, guys, I think someone left their laptop here.
Authentication required. Temporary access granted. Welcome, scientists.
Is this some kind of escape room?
Well, are we gonna do it?
I mean, we're already here.
Case File 001. An unidentified pathogen has contaminated the town municipal water supply. Location: Town Water Treatment Plant. 137 confirmed cases have been reported. Symptoms include fever, vomiting, and diarrhea. The causative pathogen remains unknown. The outbreak cannot be contained until the pathogen is detected quickly and accurately. Challenge 1 unlocked. Identify the threat. Challenge 1. Identify the threat. Recording has started. Please discuss your findings.
So we are not even looking for a specific organism yet.
No, just unknown pathogen.
Okay, but what exactly counts as a pathogen?
A pathogen is anything that causes disease. That could be bacteria or viruses, and they're found everywhere around us, so we need to detect the pathogen as soon as possible.
Which makes sense, because if we don't know what's causing it, we don't know how to stop it.
Exactly, and that's why rapid detection is such a big deal. The longer it takes to identify the pathogen, the more time it has to spread.
It could be spreading from something like the town's water supply.
Recording terminated. Uploading team response. Analyzing discussion. Knowledge verified. Microbial pathogens are disease-causing microorganisms. Rapid pathogen detection is essential for outbreak control. Challenge 1 complete. Challenge 2 unlocked. System corrupted. Repair the biosensor architecture to continue.
Wait, before we can continue, what's a microbial sensor?
Microbiology sensor is a technology used to detect microorganisms or a biological target related to them and convert that detection into a measurable agent.
So instead of waiting until we can actually see the pathogen growing, you can detect it much earlier.
Biosensors are one of the main types of microbial sensors. They use biological components to recognize targets and convert them into an interaction and signal that is measurable.
So they all follow the same basic principle. The only thing that changes is the biological recognition.
Bioreceptors are going to be the first one because it first recognizes the pathogen. Then the transducer will convert the biological interaction into a measurable signal. And then finally, the signal output will give us a result that could possibly be a color change, fluorescence, or an electrical signal.
Architecture restored. Uploading biosensor sequence. Scanning restored sequence. Bioreceptor confirmed. Target recognition begins here. Transducer confirmed. Biological interaction converted into a measurable signal. Signal output confirmed. Result may appear as color, fluorescence, or electrical reading. Challenge 2 complete. Proceeding to available detection technologies.
Why don't we just culture the pathogen?
Growing the pathogen can take out hours, maybe even 2 weeks.
And during an outbreak, waiting that long could allow the infection to spread even further.
What about PCR?
PCR is much faster and very sensitive.
But it is expensive and it can distinguish between live and dead DNA.
So positive result don't always means that it's active infection.
What are immunological methods?
These methods detect pathogen using antigen-antibody reaction.
They're simple and easy to perform, but it's too early in the infection. The immune response may not be detectable yet.
Processing. Traditional method insufficient. All available methods assessed. Traditional methods insufficient. Traditional methods remain valuable. However, their limitations reduce rapid outbreak response. New detection technology available. CRISPR biosensor.
CRISPR, isn't that a gene editing technology?
It originally is. CRISPR-Cas is actually a natural defense system found in bacteria. They use it to recognize and cut foreign genetic material from invading viruses.
So how does that help us detect pathogens?
Challenge 4 unlocked.
We have to start with the sample. If there's no pathogen, then there's nothing to detect.
Next, the guide RNA search for the matching DNA or RNA sequence for the pathogen.
Once the guide RNA finds the correct target, it activates the Cas protein.
And once the Cas protein is deactivated, it produces a detectable signal.
Detection pathway restored. Sequence confirmed. CRISPR biosensor mission successful.
First, the gRNA binds to the target DNA, then the Cas9 recognizes the target, the DNA is cleaved, and then the target DNA is disrupted. Then the reporter signal is activated.
Final challenge unlocked.
CRISPR are much faster than many traditional detection methods and they are highly sensitive.
They're also highly specific because the guide RNA recognize a precise genetic sequence that reduce false detection.
Some systems can even detect multiple pathogens at the same time.
Recording terminated. Counterargument required.
So aren't they used everywhere already?
There's still limitations. Samples need careful preparation before testing, and substances in the sample can interfere with the results.
They're also really expensive, and the technology needs further standardization and clinical approval before it can be widely implemented.
Limitations acknowledged. Future status required.
The future looks really promising. Researchers are developing portable CRISPR biosensors for point-of-care and real-time pathogen detection.
Some systems are even being integrated in smartphones, allowing results to be analyzed and displayed more conveniently.
Artificial intelligence is also being explored to help analyze biosensor data and improve diagnostic accuracy.
So our answer is yes.
But with future developments.
Decision accepted. CRISPR biosensor deployment approved with further development. Outbreak contained. Exit unlocked. Simulation terminated. Resolution file opened. Waterborne bacterial contamination identified. CRISPR biosensor detection enabled rapid containment. Mission complete. Temporary authorization revoked. Goodbye.
Oh,